have any sibling goes for it. Hold it up now, we'll start to talk about yeah, it's kind of a, she's with me for a year. Like, slowly over time, I see what my job to her. So, yeah, now that person's coming to me. I know, it's in the first class.
Okay, why don't we go ahead and get started? Nice big crowd. We're all excited for Rory, not to put any pressure on her. Anyways, as probably just about everyone here knows, today is the Dissertation Defense by Rory, and she's been a fixture around the department for a while, but is now about to make a next step in her career. And so she will present a defense of the work that she has done in her dissertation, and then we'll have open for public questions, and then as everyone knows, the audience will be excused. Her committee will remain behind and ask some additional questions, and then she'll be excused, and then they'll decide whether she successfully defended her thesis or dissertation. Most of you know Rory. She originally hails from Alaska, and she did her undergraduate work here at University of Portland as a math major, and now has been in our PhD program in the bioinformatics and computational biology track for the last several years, and so I will let her go forward.
Thank you, Bill, for the nice introduction. Can everyone hear me okay in the back? Okay, so I'm very excited to be here today and to share my dissertation work with you, so thank you for being here. Okay, so let's get started. We often talk about precision cancer treatment or precision oncology. And when we say this, what we mean is the vision that we can match each cancer patient with the best drug for them. So I'd like to go through a couple examples of what this might actually look like. So in the top panel, what we have is something called an umbrella trial. So this is a clinical trial where we have a group of patients and we have genetic sequencing information. And then we actually use that genetic information to group the patients into different subgroups. And then each subgroup gets a different drug. So you can see then that we've matched each patient with the best drug for them. So another example is called predictive modeling. So this is very near and dear to those of us on the computational side. Here the idea is that with all that rich genetic information about a cohort of patients, we can actually computationally model, or it's called in silico modeling, to figure out beforehand who's going to respond to a drug or who might not respond to a drug. So also predicting adverse effects as well. So one of my favorite examples is called repurposing. So this is more of a drug-centric perspective, so thinking about how the drug actually works. In the arm across the top here, we have the drug acting through one mechanism of action, and we use that to treat a group of people. But if we know another mechanism of action, for how that drug works, then we may actually be able to treat a whole second group of people, potentially with a different disease entirely. And then lastly, because this is very relevant for designing cancer treatments, we have combination drug design. So now imagine for one patient we're looking at their genetic information, and we actually see that they have multiple genetic aberrations, and so they might need 2 or more drugs to actually design the best treatment for them. So this is the vision, and this is where we'd like to go, but we're definitely not there yet. There's a lot of barriers that we face into making this a reality, both on the research side and in the clinic side. So I'd like to go through basically the questions that I have asked in my research and give you kind of a roadmap through my work and in the problem space that I work on in this area. So it starts with how our drugs work. So we need to know how our drugs actually act in order to be able to predict what they're going to do. So we're going to be talking a lot about drugs and targets, or the entities that drugs actually bind with. And we need to know not just how our drugs interact with their intended targets, but also their off targets, or what we didn't intend to hit. Then we're going to scale up a little bit and think about putting these targets into greater biological contexts. So all these molecular entities work together in biochemical pathways inside of our cells. And so now we want to be thinking about how the drugs interact with these pathways. So that's thinking about what does our druggable space look like for a set of drugs, and especially thinking about what the coverage for our drugs is in a disease or cancer-specific manner. Now that we've moved into the pathways, we really want to make sure, we want to stop and assess, is this unit appropriate for assessing drug response? That's our endpoint that we care about. Is the patient actually responding to the drug? And moving a little bit beyond that, we're going to talk about some modeling work to think about actually what happens to these biochemical pathways inside these cells, and thinking about the impact of genomic perturbation. So when we have mutations, what's the effect on those functional pathways? And then in parallel, we're also going to be thinking about the impact of drug perturbation. So kind of these two arms going down both sides. And then we're going to close with bringing it together to think about what do we still need to bring these two aspects of the modeling together so that we can go forward with this vision of actually matching patients based on their mutation information with the best drug for them. So we're going to start off talking a little bit about drugs. So many of you might have heard this before, but the dirty little secret about drugs is that we don't know how they work, actually. We have mechanistic information for very few of our drugs. So just to kind of set this up, I want to talk about some terminology. So when we say primary or intended target, we're referring to the entity or the receptor in the cell that the drug molecules actually bind to. That's how the drugs work and exert their action. We also often say secondary or off targets. So this is everything else that the drug binds to. And often when we say primary, it's talking about a target that's been implicated in a disease, so maybe what that drug was first designed for. But drugs are what we call, they exhibit polypharmacology, so they interact with many targets. And as you can see from this diagram, things have now started to get much more complicated. This drug shows that it interacts with 3 different targets. And what the arrows are representing is our confidence or our knowledge about those targets. So for instance, the yellow target has a solid line, and so that might be that we know this drug binds to this target. But then the blue and the green ones have dashed lines, and so we're not sure. Maybe only a couple studies have been conducted. So there's a degree of uncertainty about that. So from a research standpoint, it becomes really important that we have information for how our drugs work across all these targets. But we face a problem in that when we go out into the public literature or into the public domain, the information is very convoluted for drug-target interactions. And oftentimes different resources will report different targets for the same drugs. So if you're trying to pull this information together, it puts a very big burden on you to sort through this information every time you have a question about how that drug works. So in the first aim of my work, I've gone out and aggregated drug-target interaction data for about 140 cancer-approved drugs. So for these drugs, I've pulled together information from 4 different databases that are all publicly available. That means they're free for any of us to use, and put that all into a database. So this is called the cancer targetome, right? Because for all those drugs, it houses the target interactions. And what's unique about the cancer targetome is that it's actually paired with an evidence framework. So what that means is for every drug-target interaction you look up, you can actually ask the question of how confident are we? What's the supporting evidence behind it? So on the right-hand side here, I'm showing the evidence levels that we assign to drug-target information. So level 1 means that we found it in a database. Level 2 means that when we pulled it from the database, we also had a literature reference. So there's been an article published somewhere describing that interaction. And then level 3 means we have that information, but then we also have a binding assay value. So that means that we have the record reported of an experimental binding assay that actually tested that drug and target binding. So you can imagine that this becomes very important when you go to see what targets your drugs binds to, because you'd like to know what the strength of evidence is supporting that interaction. And so having all this information together allows us to actually start to prioritize or we get through the weeds in all of this data. So we found very nicely that it actually is important to aggregate from multiple resources. So this plot is just showing that the aggregation improves our coverage. So the first 4 bars in each plot are showing you the coverage we get for the drugs, for the targets they bind to, and the unique interactions. And what's important here is the dark blue bar, which you can see is higher just a little bit for the drugs, quite a bit for the targets and the interactions, showing that each of these databases actually contains different information, and when we pull it all together, we get the best picture. When we look at the breakdown of our coverage by evidence level, things start to get a little more interesting. So starting with the yellow bar is where we actually allow for all interactions, so any drug target information supported by any evidence of level. And then within each panel, as you go to the right and you get to the darker blue, we're requiring stronger and stronger evidence. So as we get to the blue bar, we're actually requiring that we have a binding assay reported, and then as it gets darker, we're saying that that binding assay value has to be lower. And what that reflects is that there's a stronger binding relationship between the drug and the target. And so just to tell you these levels a little bit, so the light blue is at 10,000 nanomolar for the binding assays. So that's a general cutoff we use to say that there might be activity between this drug and this target. But then all the way to the darkest blue here is down to 100 nanomolar, and that's really the space we need to be in to think about whether these drug-target interactions might be relevant for the human body, right? So not just activity, but whether it's potentially going to be clinically relevant. Okay, so we started out talking about an example of thinking about our intended targets, right? So the drug imatinib or Gleevec is a breakthrough targeted therapy for chronic myeloid leukemia, so it was really a game changer for that disease, and it's one of the best examples we have of having a very, very strong primary target. So it exerts its action through the target ABL1, which is shown in purple here. So we can see it up in the top left corner. The way this plot works is each of these panels is showing you a different type of binding assay value. So there's 4 different types here. They're just 4 different ways of getting at that binding activity. And what's important here is that we know imatinib binds to ABL1, and so we'd like to be able to see in all this data that we collected, is that showing up? And sure enough, we can see that that dark purple shows up in all 4 lines of evidence. And it's very, very far to the left, so it has a very, very low or very strong binding assay value. So I will note that there are additional targets that this drug binds to. You can see all the other colors. Each color is 1 target. So we can see PDGF beta and PDGFR alpha, as well as DDR1. These are also targets that this drug binds to. But ABL1 is supported across all 4 binding assay types. So now we're starting to see how it's really important to have all this type of information actually behind the drug-target interaction to get more detailed information. So I also mentioned this idea of these secondary or these off targets. So this is here we are now with the blue and the green. And just to illustrate why this is so important that we keep this on our radar, I'd like to show another example. So this drug, vandatinib, here I'm showing you the same range of binding assay values that we saw before, but now it has 26 different targets that it binds to. So that's a lot of off-targeting, and we'd potentially really like to know that when we're thinking about using this drug. Interestingly here too, so in the top panel, you see a lot of blue bars for EGFR, but we also see ABL1, KIT, RIPK2, and RET. And then in the second panel, we're seeing a lot of red bars. That's KDR, We do see one bar for EGFR, FLT1, and SERC. And what I'd like to note here is that in this case, for this drug, the panels are actually providing us with different information. So if you were just to look at the top panel or just to look at this bottom panel, you would have a very different view of the targets that this drug interacts with. And so this shows us that it's really important, again, to have all that information, because if we take just a limited view, we're not going to have the full targeting profile. Okay, so now we're going to scale up a little bit to start think about those biochemical pathways and druggable space. So what I'm showing here is the orange diamonds are drugs, and they're interacting with a biochemical pathway, which is called pre-notch transcription and translation. It's in the yellow there. So each node here is one pathway, and as we go up farther, they're hierarchical relationships, so child to parent pathways. So the orange, the drugs are interacting with this pathway through the targets within the pathway. And to introduce some terminology here, light pathway is a pathway that the drug interacts with potentially, whereas a dark pathway shown in blue is one where the drug does not interact with any targets. So in this case, these two drugs are arsenic trioxide and vinblastine, and you can see the targets they're interacting with are Jun and CCND1. And importantly, they're supported by level 2 evidence. So that means we actually don't have reported binding assay values for them, and if we were to require that rigor of information, then we would have all of these pathways go dark and potentially not be druggable. Just to put this into bigger context, so we were in that little square, but these pathways are nested and hierarchical, so they're all involved in higher order processes, and this is just to kind of give you a big picture of that. And so when we're thinking about mapping the drugs to these pathways using this light and dark framework, it really gives us an idea of the scope of our current drugs, especially when we start to think about whether patients have mutations in these pathways, if we could even get at those pathways with our current drugs. So using a total of 2,008 characterized pathways from the database Reactome, when we use all the cancer drug information we have, about 60% of the pathways are light or potentially druggable. So that starts to give you an idea of what we might actually be able to do with that set of drugs. As we require more and more strict evidence, so coming down to level 3, we're down to only 50% of the pathways. And requiring under that 100 nanomolar, so remember where we're starting to be clinically relevant, we're at about a third of pathways are potentially light. So really the picture would look something like this, where most of the pathways are still dark and about a third of them are potentially druggable with our FDA-approved cancer drugs. So this becomes important when we're thinking about a cancer-specific or disease relevance in a disease-relevant manner. So this is work from the head and neck squamous cell carcinoma group. So the PIs are Dr. McWeeny and Dr. Koulis-Martin, and I've been working with this group during my time here. And this analysis is by Gabby Chunu. And looking at enriched pathways, so that means pathways that are very important specifically for this type of cancer. Gabby found that 60% of those are dark to current drugs and 40% are potentially light. So you can see how we can really start to power the use of cancer targetome in a disease-specific manner here. So I'm going to switch a little bit because and give you some background about my second aim, but now we're going to be moving into thinking about really connecting this pathway approach to drug response. So to give you some background, in my second aim, I've had the opportunity to work with data from the Beat AML consortium. So this is a really big effort at treating a big consortium for understanding the disease acute myeloid leukemia. And so over the last several years, this effort has collected over 900 samples from patients with AML. And what's really unique about this huge effort is that they've collected both genomic information, so we have RNA expression and whole exome sequencing, but they're also conducting functional assays, so drug screens, to actually see if the patient samples, how they respond to different cancer drugs. And so a big key part of this work is to put these different types of data together in modeling and prioritization and actually nominate targets for future work, and then moving into clinical trials to find new therapies for patients with AML. So a little bit about this disease. So it's a type of blood cancer. It's seen very little progress in the last 40 years in terms of treatment. One of the things about AML that makes it very challenging is that the patients from a genomic perspective can look very, very different. So in this inset here, what I'm showing is the significantly mutated genes that come up in this disease. And what you can see here is basically this characterization of what we call the long tail in cancer, where on the left side we have some genes that we see in many samples or many patients with AML, but then very quickly there's a lot of genes which only some people have. And so this can make it very difficult when we're thinking about subgrouping or figuring out the best treatment, this heterogeneity as we call it. Okay, so with that in mind, I've had the opportunity to work with about 200 patient samples for de novo AML. So that means that the AML has arisen for the first time rather than being related to another treatment, for instance. I'm using paired tumor normal samples and the mutation data. And just to give some perspectives, when we're thinking about mapping these mutations to pathways, there's two different takes that we can think about. So in the top panel, I'm showing pathway burden by gene. So here we're taking those pathways that we talked about, and we're saying what's the burden or the mutational load when we map mutations to that pathway. So you can see in the schematic there's both a yellow and a blue node that we're seeing a mutation in that pathway. Then we ask the question basically if there's more mutations in that pathway, more genes mutated than we would expect at random. So is it very interesting that this pathway, and is this pathway very important? But an alternative way to look at this is to actually think about pathway burden by cohort, where instead of mapping the genes, we think about the number of samples that have an alteration in the pathway. And this allows us a slightly different take. And importantly, we can think about the example where we might have a small number of genes affected in the pathway, and that wouldn't necessarily be enough to show up in the first approach. But if there's a lot of samples with those small number of genes, we really want to get at that information. So this is the results of looking at the enrichment from the first perspective. So this is basically asking the question of which pathways have more genes mutated than we would expect at random for this disease AML. And so there's three pathways that came up: chromatin organization, collagen biosynthesis, and RNA polymerase III transcription. And they're just shown in red to show that they passed the statistical significance here. So to compare this to the other perspective, we can also think about the pathway burden by cohort. And so here we see a lot more pathways that are showing up, really highlighting the importance of taking a different perspective for that pathway burden. So here there's about 46 pathways that have a large number of samples that all have alterations in the pathway. And many of them make a lot of sense for what we know about AML. So signaling by B-cell receptor, we've got signaling by SCF-KIT and other interleukin signaling, which is where FLT3 is homed, which is a very important gene in AML. So this tells us where, now we're thinking about mutational burden for pathways, but we really want to bring that home and connect it to actual drug response. So when we assess whether there is a relationship between having mutations in a certain pathway and actual drug response, we find that there is in many cases. So this is what's called a volcano plot, and Each one of the circles that you see represents a single test between a drug and a pathway. And so what we did is for our cohort of samples, we basically have split them into two groups for each of these tests, where you say if they have a mutation in the pathway versus if they don't, and is there a difference in their drug response. So one of the big circles at the top here is sunitinib and other interleukin signaling. So that's where FLT3 is, and FLT3 interacts with sunitinib. So this was like a really great positive control. We have a lot of the FLT3 pathways and FLT3 inhibitors showing up here. So, and I should have said this, but as we get to the top of the graph, the interactions are more significant. And then as you go to the left, we're looking at interactions that are sensitive, so meaning when you have a mutation in that pathway, you have a better response to the drug. On the right, we have resistant interactions, so having a mutation in that pathway means you're actually less responsive to the drug. And to show you an example of one of these a little bit more in depth to help make it clear, so this is just one of those circles that we saw in the last plot, and we're kind of basically zoom in a little further. So each dot here is now a sample. On the left side, we have all the samples where they don't have mutations in this pathway, which is signaling by SCF-KIT. And on the right, we have all the samples where they do have a mutation in the pathway. And so there's actually a significant difference between these two box plots. And if we look at all the samples where they do have mutations in SCF-KIT, we can see that they all have different genes mutated in this pathway. So this approach of looking at the pathway view actually allows us to group samples and aggregate across many different genes that are mutated. And then I'd just like to call out that in blue we have NRAS mutated, and then we also have KRAS, and PTPN11 is in purple. So there's really a whole spectrum of genes that are in this pathway and potentially driving this resistant interaction, because these are samples that are showing higher AUC or less response to this drug, which is NVP-ADW742. It's an inhibitor. So, so we see these genes, and so the next question is really to ask if this gave us more information than looking at individual relationships between the gene and the drug. So this is also a volcano plot. It's a little bit different because it's just assessing the difference between each gene and the drug response. And so if we come into the right corner in blue here, what you can see is that NRAS and KRAS were both the genes that came up as being associated with resistance to this inhibitor. And so those were two that we saw in our pathway, but in our previous slide we also saw many additional genes that aren't showing up as individually associated with this inhibitor. And then I'll just say that this is work by Dr. Beth Wilmot, Dr. McWeeny, and also Daniel Bottomley as part of Visome for the Beat AML work. So in the inset, that was the box plot that we just looked at, so that interaction between the pathway and the inhibitor. And that's what we get when we just basically map mutations to the pathway and we ask, is the pathway mutated or not? But now we really want to dig in a little deeper and ask, how do mutations in the pathway, right? We saw those different genes are mutated. How do they actually affect drug response differently? And so we need to think about how these genes might be having different effects in the pathway. And so we're going to start to go into pathway impact and the modeling work that I've done for this. And just as a quick intro, I'll be using a type of modeling called probabilistic graphical models. And these very nicely allow us to convert those biochemical pathways, so represent them as mathematical models. And we have a suite of algorithms called message passing algorithms that allow us to basically impact or perturb one node in the graph and propagate that impact elsewhere. And this allows us to actually assess impact of mutations across the pathway. So a little bit of an overview, we're going to think of the null pathway model or no perturbation. So that's just how that biochemical pathway functions as it is. And then on the left-hand side, we're going to think about adding the perturbation of a mutation. So when we have mutations in our genes, how does that affect how that pathway would normally function? And then on the right-hand side, we have the pathway model with drug perturbation. So thinking about when drugs bind to targets within that pathway, what is the resulting perturbation in that case? Okay, so each pathway in reactome, so each of those biochemical processes is represented as what is called a factor graph, so that's shown here. Factor graphs have two types of nodes. So the circle nodes where you see X1 and X2 are called variable nodes. They each represent one of the entities in the pathway. Variable nodes can have two states, 0 or 1, and they can be not impacted or impacted. And what's important about the type of modeling we're doing is that each of those states actually is associated with a probability. So we're really thinking about the probability that X1 is impacted or not impacted. And then the second type of node in this graph is shown as the squares, so FA, FB, and FC. And those are factor functions that basically describe the relationship between nodes. So if X1 is very impacted, will X2 be impacted? So the way this works is that we have some observed entities. So these are places in the pathway, entities in the pathway in where we actually see mutations in the gene. So now it's shown in blue. And then we have this message passing algorithm that actually allows us to propagate the impact through the graph to the neighbors so that we can see the impact on the rest of the pathway. And then for each entity in the graph, we're actually quantifying, you can think of it as the final probability of whether it's going to be in the 0 or the 1 state, and that's just known as the entity beliefs. And we repeat this process many times until we basically arrive at one not changing belief, or that's also called convergence for each entity. So we're running this for each sample, gets their mutations loaded onto the pathways, and then we're looking at the impacted pathways per sample. So on the right side is what I showed you with the observed mutations, and then I just wanted to be kind of clear about then we can actually compare it to what we get from running the pathway model without any mutation information. So comparing mutation to null. And this allows us for each entity to actually come up with a log odds ratio. So basically just what I'm showing here is X1, we see what we get for the belief in the mutated state, and we're going to compare it back to the probabilities that we had in the null state. And then we do this for each entity in the graph, right, so that we know where we stand. And then at the end for the whole pathway, we're actually going to summarize the pathway impact using the average of the log odds across these entities. So here's the result of the mutation impacted pathways for the AML de novo samples. So for 187 samples we are looking at. So what you can see here is each circle is one pathway that the modeling says is very impacted based on mutations that we see for the cohort. So up at the top you can see neutrophil degranulation actually has 60 samples that are impacted for this pathway. And we also see signaling by type 1 IGF-1R, generic transcription. Other interleukin signaling is where FLT3 is, so that's one that we would expect to see for AML. And then I'll also point out this one here, G-alpha-Q signaling events, which is involved in GPCR signaling, because we're going to come back to this pathway as it has a very, very high impact compared to other pathways. It's much farther out here. So remember at the beginning we talked about the parallel approaches. So we're also going to be running this modeling for the drug perturbation. So now instead of the blue mutations that I showed you, we're actually thinking about the drug binding to the target and propagating the effect through the graph after the drug binds. And so very nicely here we actually have all this cancer targetome binding information to use from the first aim. And so when we require level 3 or binding assay evidence, we can actually use the strength of the binding assay to quantify the effect that we're having of the drug binding on the target. So this is similar to the graph we saw before, only now it's showing drug impacted pathways. So there were a total of 32 drugs that we had in the Beat AML drug screen, and we also have information on them in Cancer Targetome. And so these are the impacted pathways for that set of drugs. A lot of them are kinase inhibitors, and what we can see is that there's the RAF-MAP kinase cascade, NGF signaling, signaling by ERBB2, SCF-KIT, which we talked about, and RET. And so this gives us a perspective of the pathways that are affected by drugs now, so that second arm. And then also here's our G-alpha-Q signaling that we're going to come back to. One other thing that I'd like to note here is this is a different view on those same drug-impacted pathways, that the drug binding can actually have a very different impact on the pathway. So the size of the circle here is indicating the number of drugs that impact that pathway. And if you look at the top, our friend G-alpha-Q signaling has 3 drugs that interact with it, and they all have a very similar effect, a very high impact. But if you look at RET signaling, which is the first red circle from the top, so what the bars are showing you is the drug that had the minimum impact and the drug that had the maximum impact. And so we can see there's a very wide range in the drugs interacting with RET signaling. There's actually 20 different drugs interacting with this pathway, but the degree of impact that they have looks to be very different from the modeling. Okay, so I'd like to provide some discussion on how we might begin to unite these two modeling approaches, because that's really the vision of where we'd like to go, is how can we use individual patient level mutation information and figure out the best drug for that patient. So this is a version of the blue graph that we just showed, only now instead of the dots being blue, I've actually colored them to indicate whether they're drug impacted or not. So we're starting to intersect these two approaches. So the darker red it goes is the more impact that the drug is predicted to have on the pathway. And so now we're going to come back to our example of G-alpha-Q signaling. So this pathway has both a high impact from the mutation and from the drug perspective. So we'd like to look a little more closely at examples like this. So there are 12 samples in this group that have mutations in this pathway. And so what this graph is showing is on the left, it's showing the genes that each sample had a mutation in. Each circle on the graph is one sample. And so what we can see is while there's 12 patients that all had mutations in this pathway, they all have mutations in very different genes, right? So again, highlighting one of the advantages of the pathway perspective is bringing together many of these different genes. Interestingly, from the modeling, all these genes were predicted to have a very high impact on this pathway. And so if we look from the drug perspective now, there's 3 drugs interacting with this pathway that also have a very high impact. So bortezomib is in red, idelalisib is in green, and sorafenib is in blue. And now from this perspective, it's showing the targets in the pathway that the drug interacts with. So given that we see that this pathway both has a high mutation impact in individual patients and also potentially high impact from drug, we'd start to look and see if there's actually a difference in drug responses for these patients. So just as one quick example, so this is showing the distribution of the drug response for sorafenib, so one of the drugs that I pointed out to you. So each sample here is shown as a dot, and if that patient has a mutation in the pathway that we've been discussing, it's colored, and we can see the gene listed on the right here. And so we can see that 3 of these patients who have a very, very low AUC, or more sensitive to the drug, I should say, do have mutations in this pathway. So this is, and then I'll show one other way to look at this. And so now we're actually looking at it by each sample. So each sample is one pane, and we're seeing their response to the 3 different drugs that the modeling shows interact with this pathway. And you can see that several samples have sorafenib, the blue, very low. That's who we saw in the other plot. The idelalisib in green, and then bortezomib in red. And so this is just one way of showing how we might unite these two frameworks to really start to think about bringing together the mutation and the drug impacted. There's definitely much more work to be done in this vein. This is the start. So for instance, going in more to seeing what other gene mutations that these samples have in other pathways, because right now we're looking at just the G-alpha-Q signaling. But this starts to show how we can use the modeling framework to potentially prioritize pathways of very high interest for our samples. So in working towards a unified model of both mutation and drug perturbation, we've identified some key areas, definitely for the next iterations. So the first is curation of pathways. So I've been talking a lot about these biochemical pathways and coming from Reactome. A really key piece of the type of modeling that we're doing, this impact modeling, relies on those pathways being very well connected. So what I'm showing here in the left corner is some entities in a pathway have dashed lines, right? And then we also have one that's actually separate. So in pathways where we see something like this, where the connections are incomplete, it's very difficult for us to model impact because we're not going to be able to propagate fully. As a second needed component is the stability of pathways. So I haven't gone into this very much in today's talk, but I will just mention that that message passing that I showed you is not necessarily guaranteed to find a final result or guaranteed to converge. Especially in the case of pathways with a high number of loops, we found that it might not be possible to get to those beliefs. And so that's definitely something to keep in mind going forward. And then as we move more towards understanding or towards using this modeling work with drug sensitivity, one thing we definitely have to keep in mind is the number of samples that have drug data. So we may find subgroups of patients with impacted pathways, but as you saw from our example with 12 samples, it gets very small very fast in the number of samples that we have drug data and impacted pathways. And so we need to keep that in mind when we think about going forward with potential pathways of interest. Okay, so in the first aim, we introduced the cancer targetome as a way to capture this uncertainty in drug target information and really get our hands on drug target data. Future work that I'd love to see for cancer targetome is moving to all FDA-approved drugs. And also including investigational compounds, because right now it's just for cancer drugs, and we really like to widen up our scope. There's definitely more work that could be done for the level 2 content, so that's where we retrieved an interaction and it has a reference, but could be some questions about what is in that reference and why wasn't a binding assay value reported. Level 3 metadata, so additional annotation thinking about the cell lines that were tested for that drug target interaction, experimental conditions. We'd really like to have that information to assist in further prioritization of drug target information. So we also talked about how the cancer targetome enables us to estimate cancer druggable space, right? This idea of the light and the dark pathways. And so this is really exciting to me as potentially for repurposing, especially if we have information from drugs in non-cancer domains, we could potentially bring them in cancer uses. And we looked at how the pathway perspective actually allows us to capture associations between drug response and a pathway that very promisingly look to go beyond just single gene and drug interactions. And this could potentially take us forward for stratifying patients into different subgroups. And then we talked about the modeling framework for assessing both mutational and drug impact and really working to uniting those two arms of this framework because we'd like to be able to model patient mutational data and the drug combined to figure out what drug will be best for that patient. The modeling framework has a lot of advantages, and there's a lot of ways we could take that going forward. There's definitely room for incorporating additional gene or drug target information, such as the location of the mutation in the gene. And then something definitely to think about for this modeling is that The modeling that I have done so far treats each pathway separately, but we know that these pathways actually talk to each other, so crosstalk. So thinking about how to take that into consideration as well. So with that, I would like to acknowledge my dissertation advisory committee. So Dr. McWeeny, Dr. Wu, Dr. Cohen, Dr. Bratt, and Dr. Tyner. Then it's very important to acknowledge my funding for my PhD has come from the National Library of Medicine Informatics training grant, which was really an incredible opportunity. And the Beat AML Consortium, also a wonderful opportunity to work with the researchers and work with patient data. And so I also have to thank the patients for the donation of their samples for this work. The head and neck squamous cell carcinoma group, so that's Dr. McWeeny and Dr. Koulis-Martin's group. I've been doing work with them throughout my time here. Thank you to DMICE for being my home during my time here, faculty, staff, and fellows. And some special thanks to Dr. Dion Chase and Dr. Ted Lederis, especially for early on in my program. They provide a lot of support. Gabrielle Chunu, and then also Dr. Hannah Highlander, who was my mentor in undergraduate and set me on the path for mathematical modeling a long time ago. And then, of course, thank you to my partner, Sajan, and all my family. So open up for questions. Dion.
First of all, very impressive, very nice work.
Thank you.
A couple questions. First of all, it looks like your phase 1 database looks like primarily from bench data.
Yes.
Okay. How does the bench data you found correlate with the predicted binding assays from the mathematical models that Cohort implemented?
Oh, okay. So that's a good question. So first, yes, all the binding assay information is actually from experimental tests. So just for the audience, so Dion's also referencing that a lot of these databases will have predicted binding affinities as well for drug-target interactions. We're not using those, and the model doesn't actually, we're not predicting any strengths of new drug-target binding. So that, I would say, would be a future direction to go to, because we could see impact and then see, it could be a way of providing more validation for predicted drug-target interactions. Yeah.
One of the things I think we know about cancer is cancer mutations tend to change over time. How strong is your pathway analysis to look at those changes over time?
So I definitely think that this modeling could be done at multiple time points. So if we had the mutation data from multiple time points, then we could, for instance, get a snapshot of the pathway impacts at different stages. I think it could be adapted for that. Did you have another question? Okay.
Good work.
Thank you.
Well, let's go. I
Okay.
think I may have asked you this when you defended your proposal, but the interactions are not necessarily beneficial or detrimental, and so is there a way to model that? Because it seems that might, in terms of its usefulness in identifying those drugs that could be repurposed, could do that.
So thinking about like which, and really thinking about beneficial targets, it really depends on the purpose and what we're trying to use them for. But I think you're also getting at thinking about which targets and pathways might be leading to side effects or adverse effects as well as that.
Or even things, I mean, I know there's things in pathways that actually make cancer worse from a clinical standpoint.
Okay, so yeah, thinking about how we could prioritize not just for the pathways we want to hit, but also if we want to avoid. And then potentially a future direction would be to be very strategic with the modeling for combinations of therapies, especially for tackling resistance. Mitzi.
Great job.
Thanks.
That's amazing. So if you go back to your slide about the factographs.
Yeah, sure.
Yeah. And maybe I'm not understanding it correctly, but you talk binding.
Would you like this
affinity.
one?
Yes. So when you're talking about binding affinity being directly related to.
Oh,
Is that a different one?
this one.
Yes.
Yeah.
Being directly related to the impact of the drug on the target.
Yeah.
How are you? Can that be directly correlated?
So thank you for asking that question, because for this talk it was a little bit under the hood for that. So basically for cancer targetome we have these binding assay values, so they're reflecting the strength of the interaction between the drug and the target. And then we're actually using those directly in our graph. So remember I told you that each entity can be in the 0 or the 1 state. We're actually converting that to model the impact. So if X1 is a drug target and it has a very, very high or very strong binding assay value, then we're pushing that to be with very high probability in the 1 state. And so there's actually a conversion that I didn't show here where we're actually taking the binding assay values and coding them into the impact on those nodes. Thank you. Any other questions? Okay, well, thank you all so much.
So we will leave you to
Let me ask.